Dravet syndrome is a complex, lifelong disorder characterized by frequent, prolonged, and difficult-to-treat seizures. Typically, these seizures begin in the first year of life of a previously healthy baby and are difficult to control even with multiple anti-seizure medications. However, Dravet syndrome is not limited to seizures. Children living with Dravet also experience significant learning and developmental disabilities, behavioral difficulties, and problems eating, sleeping, and walking. Sadly, 1/5 of patients with Dravet die before reaching adulthood. Dravet syndrome affects a specific type of brain cell called inhibitory GABAergic neurons, which normally modulate the brain's electrical activity. For people living with Dravet, because GABAergic neurons are not functioning properly, the brain becomes hyperactive, leading to seizures and other clinical manifestations. The malfunction of GABAergic neurons is caused by a faulty gene that disrupts the production of sodium channels in the brain. These channels allow neurons to communicate with each other so we can think and behave the way we do. If these channels are altered, so is the communication between neurons, making it hard for the brain to work properly. There are drugs available to reduce the seizure frequency in Dravet syndrome, but none address the underlying genetic cause and neurodevelopment impact of the disorder. Gene therapy is being developed by Encoded to hopefully address these major limitations. In Dravet, the faulty gene is called SCN1A. Normally, people have 2 working copies of the SCN1A gene, which together are responsible for the production of sodium channels in the brain. But in Dravet, 1 copy of the gene is not functioning properly. Think of the 2 SCN1A gene copies as assembly lines, both needed to produce enough sodium channels for the brain to work properly. If one of the assembly lines doesn't work properly, the production output is reduced by half. Encoded is investigating a gene regulation therapy that uses a vector form of the adeno-associated virus, or AAV, as a gene delivery system for a one-time local administration directly to the brain. The AAV vector is not pathogenic because the viral parts known to cause disease have been removed when making these vectors. Therapies using AAV are currently being used to treat other pediatric neurological diseases such as spinal muscular atrophy. Here is how it is expected to work. Most AAV-mediated gene therapies are designed to replace a faulty gene, but because certain genes, including SCN1A, are too large to fit within the AAV vector, Encoded has developed an alternative called ETX101. The AAV vector delivers a gene that aims to program the cell continuously to produce a specialized protein, known as an engineered transcription factor. This engineered transcription factor binds close to the SCN1A gene, increasing its activity, specifically in inhibitory GABAergic neurons. To use our assembly line example from before, it is expected that ETX101 will boost the working line to increase the production of sodium channels and restore normal functioning. In animal models of Dravet syndrome, ETX101 achieved broad distribution throughout the brain, increased the level of sodium channels, and significantly reduced premature mortality and heat-induced seizures. With our investigational gene regulation therapy approach, Encoded is targeting the underlying genetic cause of Dravet syndrome to potentially treat not only seizures, but also the cognitive, behavioral, and developmental problems associated with the disorder. Encoded gene regulation therapy aims to offer people living with Dravet the potential for long, lasting benefits with a one-time administration. To learn more about ETX101 and our gene therapies, visit encoded.com.